Okay, good day everyone. And this is the part two of this session. Um, our topic is cell. I'm still Dr. Gerald Hiron, and I'll be the lecturer for this topic. Now, for the learning object outcomes, we will be describing the general parts of a cell and its function, differentiate the process of diffusion and osmosis. We'll be comparing an active and passive transport of substances through the plasma membrane, discuss the process of cellular respiration, the process of protein synthesis, differentiate the two types of cell division. Now, let's go to a generalized view of the cell. Yeah? Each cell is living structural and a functional unit that is enclosed in a membrane. And all cells can be divided into three main parts. The plasma membrane, the cytoplasm, which contains the cytosol and the different organelles, and the nucleus, which is which is where the DNA is located. All right, this is a generalized view of the cell. We will be talking about that in a while, one by one. All right, so let's discuss first the plasma membrane. It forms a cell's flexible outer surface, separating the cell's internal environment from its external environment. Now, other function of the plasma membrane, as noted here, is it regulates the flow of the materials into and out of the cell to maintain the appropriate environment for normal cellular activity. So it filters, it regulates, no, what is needed in flow and outflow of the cell to maintain homeostasis. Okay. It also plays a key role in communication among cells and in between cells and their external environment. So, in short, for the plasma membrane, it regulates, it communicates, and it forms outer surface. Now, the basic framework of for that function uh, to to regulate its function is for you to have a good structure, okay, or the basic framework. Plasma membrane is made up of a lipid bilayer. It's actually a phospholipid. It is composed of a two-tailed layers that is made up of phospholipids, cholesterol, and glycolipids. Okay, so other than the lipid bilayer, it also contains proteins. We have two types of proteins. We have the integral proteins and the peripheral proteins. Now, when we say integral proteins, they extend through the lipid bilayer. Okay, whereas the peripheral layer, they're only loosely attached to the exterior, either exterior or at the interior surface of the membrane. But what traverses or what extends through that lipid bilayer is only the integral protein. No, that integral proteins allow small and medium-sized water-soluble materials to cross the membrane, whereas the peripheral protein, they're only attached to either an exterior or interior surface. Okay. Now, all books about cell or any anatomy and physiology books, plasma membrane, when we say plasma membrane, it is selectively permeable. No? By definition, it means it allows some substances to move in and out of the cell, but it restricts the passage of other substances, usually large substances. Okay. Now, when we say lipid bilayer, it is only permeable to water and to lipid-soluble molecules such as fatty acids, oxygen, carbon dioxide, and other soluble substances or molecules. They are not permeable to ions and large uncharged polar molecules such as glucose and amino acid. But, Doc, can this glucose and amino acid can go in and out of the cell? Some of the small glucose and small amino acids can go in and out of the cell, but with a transporter. No, but not directly through those proteins or immediately going to from outside to the inside. No, they are selectively permeable only to these ions and molecules, but for those glucose and amino acid, they actually need some transporter. Okay. Okay, this is just a schematic diagram of the plasma membrane. So I'll use this one. Okay, this is the lipid bilayer. If you notice, there is this hydrophobic and hydrophilic layer. This is the hydrophilic, and these are the hydrophobic uh, part of the lipid bilayer. Okay. Also, this is the hydrophilic. This is the hydrophobic. That lipid bilayer um, is composed of not just the lipid bilayer or phospholipid, it also contains the plasma membrane, also contains the proteins. As I mentioned, the integral proteins and the peripheral proteins. Okay, as mentioned, by definition, the integral proteins, they extend through the lipid bilayer and the peripheral proteins are just the attachment. Now look at this diagram. The integral proteins, they traverses the lipid bilayer. No? The lipid bilayer is being traversed by this integral protein. But what about the peripheral proteins? The peripheral proteins are just the attachment. It's either on the exterior side or at the interior side. Okay? Yun lang sila. Nandito lang sila sa dyan. Naka-attach. Those are your peripheral proteins. Okay. Look at this one. If this is the extracellular fluid, we have also glycoproteins and glycolipids. So when we say glycoproteins and glycolipid, sample of which is this one, glycoprotein, if this is your protein, this is your glucoside. So this is a glycolipid because they are attached to each other. But what about this is a glycoprotein. But what about the glycolipid? Glycolipid, they are directly attached to its lipid bilayer because as we all know, the lipid bilayer is a form of a phospholipid. So this is a glucose or a glyco, which is attached to the lipid bilayer. So this is your glycolipid. Okay. So it depends on where the sugar or the carbohydrate it is attached to either a protein or a lipid. They are known as your glycoprotein or glycolipid, respectively. Okay, right. So next is the transport across the plasma membrane. This is what I'm talking about. It's not usually, you can just easily go from outside to inside. So let's first define or let's discuss some of the body fluids. Now, when you say ICF, no, or the intracellular fluid from the name itself, they are the fluid inside the body cell. When we say extracellular fluid or ECF, they are your fluid that can be seen outside of the cell. No, as the name implies, extracellular, outside of the cell. Interstitial fluid, on the other hand, is an extracellular fluid between the cells in tissues. Okay, blood plasma is the extracellular fluid in the blood vessels. Okay, what about the lymph? There are extracellular fluid that is found in the lymphatic vessels, while the cerebrospinal fluid, that is the fluid that surrounds the brain and the spinal cord. So if you have a brain injury or a spinal cord injury, usually magkakaroon ng mga leakage ng mga CSF, and that is actually a cerebrospinal fluid. It's a clear fluid, okay? But if there is a blood in it, infection in it, medyo malaki na yung damage, kailangan niya na ng antibiotics to go through that infection through blood. Okay. Next, let's define solute and solvent. So this is actually basic in chemistry. When I say solute, it is any material that is dissolved in a fluid, while the solvent is the fluid that dissolves these materials. Okay, solute and solvent. All right, so let's discuss now what are these two methods for transport across the plasma membrane. It could be a passive process or a passive transport, or an active process or active transport. No? First, let's discuss the passive processes or passive transport. The substance here moves down its concentration gradient through the membrane using only its energy of motion, and we call it kinetic energy. So example of your passive transport is simple diffusion or osmosis. So that means it will just move down its concentration gradient through the membrane by energy of motion. Okay, so that's the passive transport. What about active processes? Active processes or active transport, it uses cellular energy. It is usually in the form of ATP, and that is used to push the substance through the membrane, or it is an uphill against its concentration agent. And the perfect example of an active process is active transport. It uses ATP. Okay. All right. So these are the different types of active and passive processes. Let's first discuss the passive processes. Okay. Passive processes, it is the movement of substances downhill. Now, a concentration gradient until the equilibrium is reached. Then they do not require energy or ATP. No? First is diffusion. And when you say diffusion, diffusion and osmosis are the very common to passive processes. No? But when you say diffusion, it has two. You have simple diffusion and facilitated diffusion. When we say simple diffusion, it is just a passive movement of a substance through a lipid bilayer of the plasma membrane. And I've discussed that the lipid layer of plasma membrane, if there is a transport of substances from outside to inside or vice versa, it's just a simple diffusion. No? Lipid soluble molecules such as your oxygen, carbon dioxide, nitrogen gases, fatty acids, steroids, fat-soluble vitamins, A, D, E, K, and the polar molecules such as your water and urea can easily transport from outside to inside through simple diffusion. And that is actually a passive process. Okay. Next, what do we mean by facilitated diffusion, Doc? It also a passive movement of a substance down its concentration gradient, pero meron siyang channels or carriers. Okay, there is an ion channels or carriers. And the example of their facilitated diffusion are your potassium, chloride, sodium, calcium. These are electrolytes. Your glucose, your fructose, your galactose, and some vitamins undergo facilitated diffusion for it to be transported from outside to inside of the cell. Okay? And the second type of a passive transport is osmosis. Osmosis is just simply movement of water molecules across a selectively permeable membrane from an area of a high water concentration to an area of low water concentration. So basically, the substance just transported using osmosis is water. Okay, so just take note, diffusion and osmosis are all part of a passive transport or passive processes. Now, let's move on to your active processes. So as mentioned a while ago, when we say active processes, it uses energy. It requires cellular energy in the form of an acute of ATP or adenosine triphosphate. Now, what are these active transport? These are transport in which the cells expends or there is an energy expenditure to move a substance across the membrane against its concentration gradient, aided by membrane that act as pumps. Okay? May pumps na nga, may ATP energy pa. So these are sodium, potassium, calcium, hydrogen, iodine, chloride, and other ions, so meaning other electrolytes that needs to be transported from outside to inside in the lipid bilayer. It uses pumps, it uses ATP, and if those substances use those pumps and ATP, you are in an active transport. Okay. Else, another active processes is just a transport in vesicles. Movement of substances in and out of the cells, but this time in vesicles. Okay, this is our transport in vesicles. They require energy as well that is supplied by your ATP. Now, what are the other active processes? Well, your endocytosis, they transport in the vesicles, and exocytosis. Now let's define one by one. movement of substances into a cell. Papunta sa cell from outside to inside in vesicles. First is phagocytosis. Now, this process is also known as your cell eating. This is a movement of a solid particle into a cell after a pseudopods engulf it. Perfect example of phagocytosis are your bacterial, viruses, or aged or dead cells. So if that vesicle recognizes that it is a foreign body, it's a bacteria, it's a virus, it's a dead cell, they will try to engulf it, put it in a vesicle, and then it will start to eat or cell eating. That is actually phagocytosis. Now, it's an endocytosis process. What about your bulk phase endocytosis? If there is a cell eating, there is cell drinking. So that is your bulk phase endocytosis. It is a movement of an extracellular fluid into a cell by enfolding into its plasma membrane. Some solutes in the extracellular fluid wants to go inside the cell, and that is what we call your bulk phase endocytosis. It's a cell drinking. Okay. Next is exocytosis. As per definition, it is a movement of substances of a cell in a secretory vesicles that fuse with the plasma membrane, and it will release their contents into the extracellular fluid. So, meaning, punta mo na siya sa vesicle, it will fuse into the plasma membrane, and then after which, the content of this vesicle will release into extracellular fluid. An example of which are your neurotransmitters, hormones, digestive enzymes. So these substances or molecules, once they're released into the circulation through blood, they will go now to their target organs and they will perform its function. And that is what we call your exocytosis, from inside to outside. Okay. Now, after understanding or appreciating the plasma membrane, its function, its transport ions and molecules, let's go now to the cell parts and their functions. And from this illustration, no, you've taken this up cell in your science subjects. We have the different organelles in our cell. Example of which are your endoplasmic reticulum, your centrosome, your lysosome, your ribosome, Golgi complex, mitochondria, microfilament, and microtubule. These are your cytoskeleton. Your flagellum and the cilium are actually your attachment to your cell membrane. But the main function of the cell, the main parts of the cell are your nucleus, your cytoplasm, and the plasma membrane. Okay. All right. So as mentioned a while ago, one part or integral part of the cell is your plasma membrane. They are composed of a lipid bilayer with phospholipids, glycolipids, and cholesterol as mentioned, and they surround the cytoplasm. Function, protection, transporter. Okay, it mediates the entry and exit of substances. That is what we call your selectively permeable function of the plasma membrane. Okay, now next, let's go now to your cytoplasm. Sa inside of the cell. The cellular contents between the plasma membrane and nucleus, that is your cytoplasm or cytosol with organelles. These are the site of all intracellular activities occurring inside the cell except for those occurring in the nucleus naman. Okay? The cytosol, no? Yan ang fluid inside the cytoplasm, cytosol. They're composed of water, solutes, particles, lipid droplets, etc. And that liquid, which many of the cell's chemical reactions actually occur. Okay? It maintains the shape and the general organization of cellular contents. So, ibig sabihin, what is responsible for the cell movements is actually your cytoskeleton. Okay? Yan ang responsible for all cell movements and they are found inside the cytoplasm. Okay? Now, after the cytosol or the cytoskeleton, let's go now to the different organelles found in the cytoplasm. When we say organelles, they are actually specialized cellular structure with its characteristic shapes and specific function. Now, You will have to memorize the function of each organelle that we will be dealing with. Okay, first is the centrosome. Centrosome is or are a paired centrioles plus that material. And that pericentriolar material is an organizing center for its mitotic spindle and for microtubules for cytoskeleton. For the mitotic spindle, that is essential for cellular division. Okay. Next, your ribosome. Cilia and flagella muna. Actually, cilia and flagella are motile cell surface projection with inner core of the microtubules. They're not found inside the cell, no, but they are actually they are still considered as projection. Cell surface projection with the inner core of a microtubule. So the cilia will just move fluids over a cell surface while your flagellum will move the entire cell. So also, they're talking about the movement or locomotion of the cell. Okay, that's your cilia and flagella. Ribosome. Okay, ribosome is composed of two subunits containing the ribosomal RNA and proteins. It may be free as well in the cytosol, but if that ribosome is attached, they're attached in the rough endoplasmic reticulum. They're very nice to remember. Or if they are not attached, they are called free. But if they are attached to anyone, it is attached in the endoplasmic reticulum. Okay? Our ribosome, what is the main function? Protein synthesis. Okay? That is the main function of the ribosome. Kahit free man yan or attached, its main function is protein synthesis, or it synthesizes proteins. Okay, next is the endoplasmic reticulum. They are a membranous network of folded membranes. Now, there are two types of endoplasmic reticulum. One is rough ER or rough endoplasmic reticulum. They are studded with ribosome and they're attached to the nuclear membrane. The rough endoplasmic reticulum is the site of the synthesis of glycoproteins and phospholipids, while your smooth endoplasmic reticulum is the site of fatty acid and steroid synthesis. Please take note of that. Okay? Now, the rough and the smooth endoplasmic reticulum, again, rough endoplasmic reticulum for synthesis of your glycoproteins and the phospholipids, while your smooth endoplasmic reticulum is for fatty acid and steroid synthesis. Okay, next is the Golgi complex. This is a stock of 3 to 20 flattened membranous sacs called your cisterns. And what does it do? The Golgi complex actually stores, packages, and exports proteins in and outside of the cell. Okay. Next are your lysosomes. Your lysosomes are your vesicles that is formed from the Golgi complex contain digestive enzymes. And these digestive enzymes will help the cell to digest the contents of this vesicle, digest worn out organelles, digest the entire cells, for example, and digest some extracellular materials going from outside to the inside of the cell as well. Okay. Peroxisome is a vesicle containing oxidative enzymes. So they detoxify harmful substances such as hydrogen peroxide and associated free radicals. Your proteasome, they are a tiny barrel-shaped structure that contains proteases. These are enzymes that cut protein. So in short, its function is they degrade unneeded, damaged, or faulty proteins by cutting them into small peptides. Okay. And lastly, the mitochondria. They consist of an outer and inner membranes, cristae and matrix, and they are the site of reactions that produce most of the cell's adenosine triphosphate. They are your powerhouse of the cell. They use energy. Okay, and lastly, the nucleus. They consist of nuclear envelope with pores, nucleoli, and chromatin or their chromosomes. They contain genes which control the cellular structure, and they direct most of the cellular activities using this as nucleus. Okay, so again, in summary, the parts of the cell is or are your plasma membrane, your cytoplasm, and the nucleus. Okay. All right. So one of the cellular organelles is mitochondria, right? Within the mitochondria, the chemical reactions of aerobic cellular respiration will generate what we so called ATP or adenosine triphosphate. And the function of this mitochondria is to generate that ATP through reactions of what we call an aerobic cellular respiration. They play also an important role, the mitochondria, early role in apoptosis. So basically, aerobic cellular respiration is through this mitochondria. Okay, protein synthesis. Now, in synthesizing proteins, proteins determine the physical and chemical characteristic of the cell. Okay, there are two steps in protein synthesis. We have your transcription and translation. So, by definition, transcription is copying, producing a specific molecule of an RNA, while translation is information in that RNA is translated to become a specific amino acid to form a new protein or to synthesize a protein. So before the translation happen, there should be a transcription, okay, before it goes to being translated into a specific amino acid. Okay, now transcription occurs in the nucleus. The genetic information of DNA base is copied into a complementary sequence of codons in a strand of RNA. Now, in the transcription, they are catalyzed by what we call the RNA polymerase. Okay? RNA polymerase. They must be instructed where to start the transcription process and where to end it. Now, there are three kinds of RNA that are made up from DNA: mRNA, messenger, ribosomal RNA, and transfer RNA or tRNA. That's your transcription. As mentioned a while ago, we want to synthesize more protein. So, para mangyaring ma-synthesize a protein, we need this translation. This is the process wherein those mRNA will be associated with ribosomes. Okay? Direct synthesis of a protein, they will convert that mRNA into a specific sequence of amino acid. Okay. Yeah. So in the step one, it's the transcription. No? This is the nucleus. That is the DNA. Okay. So this is the DNA. It will be transcripted into an RNA, single-stranded RNA, as mentioned. Now that single-stranded RNA will go again here in the cytoplasm, and that RNA will be copied and it will be translated to become an amino acid to synthesize a protein. Kasi when you say protein, they are building blocks of amino acids. So you have to have amino acids first or polypeptide sequence or amino acid sequence before you produce a protein. Okay? Yeah. So this is another topic under cell. It is called your cell division. It is a process whereby cells reproduce themselves. So as body cells become damaged, diseased, or worn out, they are replaced by cell division. Okay, and there are two types of cell division. The first one is the reproductive cell division or meiosis. It is a process that produces gametes. Those are your sperm cell and your oocyte. Okay, the somatic cell division naman, the cell divides into two identical cells. Okay? And the somatic cell division, usually these are the cell division per se. No? Thus, in the cell cycle, there are sequence of changes that a cell undergoes from the time it forms until it duplicates its contents and divide again into two cells. So it's a process until a reproduction happens. there are two major periods in what we call the somatic cell division: interphase and mitotic phase. So when we say interphase, this is when a cell is not dividing and the cell replicates its DNA na. Okay? When we say mitotic phase, this is when a cell is dividing and the division of the nucleus and cytoplasm is dividing also into two cells. Okay, that is your mitotic phase. Whereas in the interphase, the cell is replicating na lang its DNA. All right, so during mitosis, the duplicated chromosomes become exactly segregated, one set into each of the two separate nuclei during mitosis. So the whole cytoplasm, the whole nucleus is being reproduced or replicated into another two set of each cell. Okay. In the mitotic process, they are divided into four stages: your prophase, your metaphase, anaphase, and telophase. These are actually being tackled early on during your science subject, no? The somatic cell division. And that ends my lecture on the cell. Thank you.